I'm Renita Malhotra-Hora, Head of Marketing and Communications at SRI International. And I have with me today Mary Lanier, who is a research scientist in our Biosciences division. Hi, Mary.
Hi, Anita. How are you?
I'm great. I am thrilled to have you on the Dish TV today. There is lots to talk about. As you already know, the question that has been coming to us from everybody out there is, what is the work that SRI International is doing to respond to the COVID-19 pandemic? That's a very wide question, and
Yes,
I know you've got specific answers. So over to you.
thank you. Yes, and so just like many other institutes, universities, government centers, SRI has pulled together resources to counter this urgent pandemic need. We have many hats in the play. We have many layers that we're working on. SRI has platforms that we have been using for other diseases that we switched to use for COVID-19 responses. And these platforms help to repurpose drugs, to design new drugs, new targets. And many of the investigators have been targeting the SARS-CoV-2, which is the cause of COVID-19, the different proteins on that to try to develop new antivirals or to repurpose antivirals to target this particular pathogen. We also have groups that are more engineering, and so they've been pulling together their intuitiveness to develop platforms to test for COVID very rapidly. And so to turn around PCR tests very quickly in order to do mass testing, for example. There have been many other programs that have used novel approaches to targeting the pathogen, and these are small molecule development projects. And these are more designed to help the immune system to target and counter the COVID-19 virus. I myself am developing a COVID program here to test all these platforms. So we do have the ability at SRI to do BSL-3 work and ABSL-3 work, which is small animal work and testing of different vaccines, different antivirals in these models. And so all of those are up and running now because SRI has put in a lot of resources in order to develop those models over the course of this year so that we can help people who are in other companies that don't have a BSL-3 or do not have a facility like SRI, so we can test those products as well.
So what is it that we are actually testing, against these pathogens?
Sure. So what we're testing different drugs. So we have antiviral drugs that are being used right now for things like herpes virus, flu, influenza virus, and we're just testing them to see if they have any efficacy against SARS-CoV-2. And so we can do this in the BSL-3 laboratory. We're also testing antibodies. So we hear a lot about antibodies and if you have protective antibodies or not protective antibodies. And so we can take those antibodies and expose them to the virus in the BSL-3 and test whether or not they actually can stop the virus from infecting. And so those are some of the things we're doing in that lab.
So the objective here is what specifically? I mean, vaccines are already out or on their way to being out. So is our objective therapeutics?
We have several objectives. SRI does have a therapeutic interest because antivirals potentially can work against this virus and potentially other coronaviruses. So there's the potential there to have protection from a potentially up and coming pandemic, you can say. Now, with respect to vaccines, even though we have vaccines in the pipeline, there is still some need to test vaccines because the vaccines in the pipeline right now are very, very specific to this coronavirus. And again, there is a lot of interest to see if we can generate a vaccine that can have cross protection across different coronaviruses with the same idea that if a novel coronavirus comes, we will have in our vaccine repertoire a vaccine that can protect against even a novel version of a coronavirus.
Wow, that's a lot of information. Let's try and break it down. You mentioned these platforms and that we were using these platforms for other kinds of viral research or virus research, I should say. And we have then, since then, switched the focus to specifically to research for COVID-19. So can you explain that process?
Yes, so the platforms I'm talking about are computer intelligence platforms to help guide small molecule development for targeting certain proteins that viruses have. For example, before COVID, these platforms were used for influenza to try to generate new antiviral targets for the influenza virus. Since COVID, we have used these intelligent computer modeling programs to sort of try to identify potential targets within the coronavirus, the COVID-19 virus, that our small molecules potentially may have efficacy against. And when we identify those, we can then take those into the lab and do actual testing against the real virus to see if the model did predict a potential candidate for targeting COVID.
When you talk about computer modeling, what does that mean? Is it an AI-driven platform?
Yes, so it is an AI-driven platform because remember, viruses are biologics. So they are made up of basically carbon, oxygen, nitrogen, hydrogen like us. And so we can use computer modeling to understand their protein structures. And so different sequences can be inputted into these programs that will properly display the folded protein, for example. And then we can also add to those our small molecules, which are also biologics. And we can identify these small molecules, whether or not they have any sort of target within that folded protein that could potentially, for example, block the spike protein from being able to bind to its receptor or something along those lines.
So then you said we have groups here at SRI which can do things like PCR testing. What does that mean?
So PCR is polymerase chain reaction. And so basically what that is measuring is it's measuring the virus genome. So you're basically are able to use a instrument which can take a blood sample, it can take a sample anywhere on the body, and it could basically see whether or not the virus is there and how much of the virus is there. So it's a test that is used right now to monitor people who are infected with COVID-19 in hospital settings, as well as to test people whether or not they've been exposed to COVID-19. And it directly measures virus. And so typically your samples, for example, you'll take a swab sample. Those are those kits that people are getting at home. You take a swab sample, send it back to a lab, and they do a PCR test on that swab sample to see if you have any virus present. And so at SRI, we have engineers that are trying to develop a handheld device. So you can take a swab sample and you can put it into the handheld device in real time and get an immediate response as to whether or not the virus is present.
This work that we're doing here at SRI, how similar or different is it to the work that's being done out there by other researchers?
A lot of people are looking into antivirals. A lot of people are looking at repurposing of drugs. We have the added ability to test those with the live virus, as I mentioned before. We have the ability to help other companies test what their novel products are against the virus as before. Some of the things I think that SRI is positioning itself for is to be able to have a more rapid response if something else were to come up. So, and in addition, because of coronavirus is potentially going to have another pandemic, we've now seen 3 major human infections come from this family of viruses. We are definitely in trying to develop a more universal approach to treating coronaviruses. And so I'm very invested in trying to figure out how we can develop something that will target all coronaviruses. And a lot of places are going to be doing that too. But I think we are positioned because we can work with all of the highly infectious coronaviruses that are present, which a lot of places cannot. So to work with SARS-1 and to work with MERS, you have to have a CDC-approved BSL-3, not just the BSL-3. So, and we're positioned so that we can work with those viruses.
From the sounds of it, what we're doing and what you are doing very successfully is really combines a mix of traditional chemistry and AI or machine learning. Is that accurate? What am I missing?
No, that's accurate for the drug development, absolutely. But it's also classic virology and it's classic immunology combined. And what combines the two is vaccinology. And so it's bringing all of those together to try to not only understand more about this COVID-19 virus, but to also be prepared for the next potential coronavirus.
Mary, thank you so much for joining us today. That's Mary Lanier. She is a research scientist at SRI Biosciences.
Thank you.